Hello everybody. This is going to be our last module that talk about microscopic examination of the brain. And in this module, we'll particularly talk about how does immunohistochemistry work and how to apply that. Okay, what is immunohistochemistry? There are three parts of this long terminology. First one is immuno. In this technique, we use the antibody, which is a component of our immune system. That's why the immuno is there. And then histo means tissue. We already talked that. And then we have to use a dye to visualize the subject that we want to see. And we have to use chemical reaction to make this dye. That's why the chemistry is in there. And in the last two modules, the technique that we covered didn't use the antibody. So that is called histochemistry. Before we talk about immunohistochemistry, there are some concepts that we have to understand. First one, you have to understand that one type of antibody only binds to one type of target. So there is a terminology for target, which is called epitope or antigen determinant. And what is an antigen? Antigen is a foreign object that enter our body that can cause an immune response. And most of the time it is a protein. And epitope is actually a part of antigen. Look, this is a big antigen that is a whole protein, and there's many epitope on the surface of the protein because a protein is a long chain of amino acid, if you remember that from your biology class, and then this long chain amino acid will fold into 3D structures, and some of the structure will expose on the surface, and that will become the binding site for an antibody. So if this is an antibody, this antibody will bind to this epitope. And epitope and antigen is really not the same thing. Somehow they get used interchangeably, which is not right. Okay, here I said epitope is a part of the protein sticking out on the 3D structure of a protein. So there are so many epitopes and they're all different. Okay, so epitope is also called antigen determinant. Okay, here's some questions to help you to understand these concepts. First one, how many type of antibodies do we have in our body? The answer is countless. We have a lot, and we are constantly making new antibodies because our bodies constantly encounter new antigen. And then a vaccine is actually an epitope from a virus protein. So when you have a flu shot, they actually inject a very small piece of certain flu protein to your body. So you are going to have a immune response to generate some antibody. Okay, here is a question. What is a good COVID-19 vaccine? It is a piece of epitope that we'll inject to our body so we can generate a effective antibody and have a least side effect. What do I mean by effective antibody? Because certain epitope, if we inject to our body, we're going to have some immune response, we're going to have some antibody, but this antibody will only bind to the epitope without taking further actions, which means they don't kill the virus. They just bind the epitope, and it's not very effective. Okay, here's the molecular structure of antibodies. So basically, antibodies are proteins, and then after they have this 3D folding, they will have this Y-shaped structure. And this Y-shaped structure will be divided into two regions. So the variable region, the blue color region, is antigen binding site. So this region will decide which epitope this antibody will bind. And then the constant region, which is this yellow part, will decide which mechanism to use to destroy the foreign object, the antigen. And then I want you to think in your body, you have a lot of different type of antibodies, and then some of them will share the same destroy mechanism. So which means their constant region will be the same. And then their antigen binding site will be different. They will bind to different epitope. It's like when you have spoon, you have forks, you can pick up different food. And then you can have other type of antibody have different destroying mechanism. And among this group of antibody, they combine to different epitope. Okay, here's how immunohistochemistry works. This green part represents the brain samples that we've been using in last two modules. They are prepared exactly the same as before. And actually, you cannot see individual proteins on the brain slice. I draw these proteins big because I want you to understand the concepts. In the real world, you cannot see them even under the microscope individually, right? And then each protein will have many epitopes. So these things sticking out, they are their epitopes. They also have epitope underneath of them. I didn't draw that. And these epitopes offer a binding site for antibodies. And here's our antibodies. And they will only bind to the epitope that have a 3D structure matching with their variable end. So you see these two epitopes, they can bind, but these things, they don't. And this antibody comes together with some dye molecules. Can be a fluorophore, can be a pigment. In this situation, that's fluorophore. You see this antibody was already have 3 fluorophore on it. And this one has another 3. And these antibodies will also have fluorophores. So when we incubate the brain slice with these antibodies, we will add a lot more antibodies in there because we want all the bindable epitope have a antibody binding to it, right? So we're gonna have a lot of extra, and these extra antibodies will be washed away. We're gonna use PBS to wash them away. Okay, when the antibody all find their matching epitope to bind, and all these free lonely antibodies are washed away, we'll be ready to put this brain slice onto a glass slide, and then we're ready to take a look the sample under the microscope. Under the microscope, we cannot tell which protein or which epitope was labeled by these fluorophore conjugated antibodies. What we can tell is we can tell, okay, so this region was stained and this region is not. And if the cell of our interest is large enough, we can tell, okay, so this cell is stained and the other cells are not. And that because both fluorophore and light microscope, they have detect limit, right? Both of them are 250 nanometers. So anything smaller than that, we cannot tell the physical boundary between them. And this one antibody binding to the epitope is called direct immunofluorescent. And there is indirect immunofluorescence, which include another antibody will bind to the first antibody. So this antibody will call it primary antibody, and this one is called secondary antibody. So what is the benefit of involving a secondary antibody? The answer is you can have a stronger signal. In this situation, you only have space for 3 fluorophore molecules. Fluorophore molecules are very small, but still they take space, right? You don't have enough for more. If you need 6, 7, 10, then this is how you solve the problem. You will have limit space on the constant region of this primary antibody, and you will use this region to bind another 2 more antibodies. And that will give you more space for fluorophore to attach. In this situation, you have 6 space. So obviously, this type of staining will give you more signal. And then if you want even a stronger signal, you can involve a HRP mediated binding, which means you have primary antibody, secondary antibody, and then instead of having a fluorophore attached to your secondary antibody, you can attach biotin, here this block, and then you attach HRP. Because biotin and HRP, they are even smaller, and then the signal is stronger. So you can choose direct fluorescent, indirect fluorescent, or this HRP based on stainings. Depend how strong you want your signal to be. As far as I know, direct immunofluorescents are really not commonly used because first of all, it's expensive, and second, the signal is really weak. And most of the experiments, they require stronger signal than that. So people will use indirect immunofluorescent way better than this one. And about this HRP-based stainings, you don't need a fluorescent microscope to see your results because the HRP is a pigment. You can see that under the light microscope. However, these techniques require longer staining steps, and there's a long list about the details you have to pay attention when you're using this technique. So we're not going to use this technique in our lab, so I'm not going to focus on this one. Okay, let's talk about where do we get these antibodies for this indirect immunofluorescence, because we're going to use these techniques in our lab. So you already know that these fluorophores are chemically synthesized. And where do we get these antibodies? So to make primary antibody and secondary body, we need animals to help us. There are 3 animals that involved. And these 3 animals has to be 3 different species. And by injecting foreign object to different animals, we can harvest different antibodies. It's kind of like gave immune shot to different animals, and they will generate antibody for us. Okay, let's take a closer look. So first one, from the animal that we will study, and this animal we call them animal 1, we'll isolate epitope. And then we'll inject this epitope to another animal, which is going to be a different species from our animal 1. Okay, and because this epitope functions as a foreign object to animal 2, this animal 2 will develop a lot of antibody against this epitope. And then we're going to purify or isolate this primary antibody from this animal 2. Then this primary antibody was made. Then we're going to use a small portion of this primary antibody to inject to another animal, and this is animal 3. And in this animal, this primary antibody functions as a foreign object, right? And this animal 3 will develop some antibody, and that's going to be our secondary antibody. Then we're going to get the blood from the 3rd animal and then isolate or purify the secondary antibody. And then the last step is we need to conjugate a fluorophore to our secondary antibody. And this is also a chemical reaction. So here is a conclusion. How many species totally involved in this process? The answer is 3. So the first one is the studied animal. In our situation, is going to be our mouse, right? We study mouse brain. So this is our animal 1. And then animal 2 is the primary antibody was developed. And then animal 3 was the secondary antibody was developed. Now, why do we have to use 3 different species? Because if you inject the same protein to a same species of animal, you are not going to have very strong immune response, and you are not going to get that much antibody. Is that right? And then if you do have to use 3 same species, will you have antibody? The answer is yes, but a very limited amount of antibody. Okay, let's talk about the epitope examples. So there are some epitope you can only find in neurons, and because of this, we'll be able to use immunofluorescent to label neurons. So there are 2 of them. One is neuin. It's also called neuronal nuclei. This is actually a protein name, and there is a multiple epitope on this protein, and all the epitope on this protein are called neuin epitope. And for short, people will just call that neuin. And actually, neuin is also an antigen name. So that's why people use antigen and epitope interchangeably. And another epitope you only find in neuron is MAP2. MAP2 is also a protein name, and on this protein you have multiple epitope, and they all called MAP2 epitope. And when we are talking about neuin stain, we mean to stain neuin containing cells with particularly antibodies. So basically we're staining neuron if we are staining neuin, because neuin only exists in neuron. And another example is astrocyte. So there is a protein or epitope called S100 beta. If that's the epitope name, that's a multiple epitope. And that is a calcium binding protein, and then the GFAP. We're going to use GFAP staining to label our astrocyte in this lab. And then the oligodendrocyte progenitor cells, there's a neuronal glia antigen 2, that is the antigen name, and also multiple epitope names. And the epitope only find the microglia cell. It is IBA1 or this one. Okay, now let's take a look at the antibodies that we're going to use in this particular lab. So we're going to label 2 epitope. or two antigen. One is neuin, the other is GFAP. And by labeling neuin, we're labeling the neuron, and by labeling GFAP, we're labeling astrocyte. So the animal that we're going to study is mouse. The brain slice was from mouse, and the primary antibody for neuin primary antibody was developed in rabbits, which means somebody takes some mouse neuin protein and inject it into a rabbit, and this rabbit will develop some antibody, and this antibody is called rabbit anti-mouse neuin. And for GFAP, that primary antibody was developed in chicken, so that's called chicken anti-mouse GFAP. And then the secondary antibody for neuin was developed in goat. So this secondary antibody was called goat anti-rabbit antibody. And then the GFAP was developed also in goat. So it's also called goat, but anti-chicken secondary antibody. And then the fluorophore that was labeled or conjugated to the secondary body was Cy3 for neuin secondary antibody, and Alexa 488 for goat secondary antibody. And then I have a spectrum here, light spectrum, and then I have some questions for you. So the first one is, what color will GFAP look like after staining? More precisely, I should ask you, what color will astrocyte look like after our staining? Because GFAP stays in astrocyte, and GFAP is too small to see individual of them, right? Okay, let's take a look. How do you decide the color? So for GFAP, we're going to have primary antibody bind to them and secondary antibody bind to your primary antibody, and this secondary antibody has a fluorophore. And then the fluorophore have excitation light of 490 nanometer and emission light of 525 nanometer. So the color you are going to see is going to be your emission light, which is going to be 525. So that is going to be somewhere in here. So GFAP or astrocyte is going to look like green under the microscope. And the excitation light we use to visualize our GFAP staining is going to be 490 nanometer, which is going to be here. This light is going to be cyan. And what color will neuin, or I should say neuron, looks like after our neuin stain? The answer should be this color. And what is 568 is? 568 is somewhere in here. It's yellowish green, right? However, our microscope doesn't have a emission filter around this. So we're going to have some color look like orangish yellow for neuin stain. Okay, here come another question. What if instead of using goat anti-rabbit and secondary antibody, I use the donkey anti-rabbit antibody for my neuin stain? Are you going to have the same result? The answer is yes. Why is that? Because even this antibody was developed in goat, it's still going to bind this primary antibody, and because this secondary antibody was conjugated with Cy3, so you are going to still use the same excitation light and see the same emission light. So you are going to have the same result if you use this antibody instead of having goat anti-rabbit secondary antibody. Okay, how about this? If you are using goat anti-chicken secondary antibody here, instead of goat anti-rabbit or donkey anti-rabbit secondary antibody, are you going to have some result? The answer is no. In this situation, you are not going to label any neuron. Why is that? Because this antibody will bind to chicken primary antibody. They are not going to bind your rabbit primary antibody. Okay, let's talk about other things to consider. When you are doing immunohistochemistry, there are so many steps that you can go wrong. It's not that easy. This lab, I make things look very easy because I did a lot of pre-experiment, and there's a lot of behind scene I didn't tell you. First one is polyclonal or monoclonal antibodies. When you are buying antibodies for your stain, when you go to your graduate school, you will see these two words. And what they mean these antibodies are made in different process. So monoclonal antibodies was made in purified cells. It's made outside of a body of an animal. And then polyclonal antibodies was made inside animal. And usually mono antibody will have less unspecific binding, less off-target binding. And poly will usually have more unspecific binding, and accordingly, poly will be cheaper and mono will be more expensive. That's in general. For certain antibodies, you probably have the same price for either mono or poly. And then next one, what is Triton X100 for? You are going to need Triton X100 in your staining. And why do you have that? Because this is the detergent. It's the detergent like the shampoo that you're using. That's the detergent. So what is a detergent can do? They can make holes, little small holes on the surface of the cell, so the antibody will be able to enter the cells. So if you didn't have this Triton X100 in your antibody solution, you will have less stainings. And then what is the blocking reagent for? You are going to have blocking reagent in your antibody solution. I already added in there. So what is the function for that? Let's go back to this slide. So if you have your brain slides here, there's a lot of epitope. And this is the epitope, is the target binding. That's the one you want. And there is another epitope. It's not 100% matched, but there's like some weaker match. They combine here. This is called unspecific binding. You don't want this to happen. So you want to have some blocking agent to block this unspecific binding. And the controls. So when you go to graduate school, when you're doing the immunohistochemistry staining, every time you buy a new antibody, you want to test the controls. You can have 3 different controls. Tissue control, which means you don't add any antibody. And primary only control means you only put primary antibody in there. Secondary only control is you only put secondary in there. You don't have a primary antibody in there. And for all these 3 controls, you should never see any staining. It's the same as your experimental image. Okay, here's another thing to consider. I said different astrocytes have different levels of GFAP. So GFAP is a protein. It makes sense. Different cell will have different levels of certain proteins, all right? Okay, so here comes the question. After our immunohistochemistry staining, which kind of astrocyte will look greener? The astrocyte with more GFAP, less GFAP, or no GFAP? The answer is more GFAP, is that right? Because the more antigen you will have, the more epitope you will have. And then the more antibody gonna bind to this astrocyte, and that will attract more secondary antibody. And then the secondary antibody was labeled with fluorophorate. That's why this one will look greener. And if astrocytes don't have a GFAP, these astrocytes will not be even stained. So I want you to keep in mind, not all the astrocytes will be labeled after the GFAP stain. Now let's talk about the applications of immunohistochemistry. There are many applications. You can use that to determine if a tumor is benign or malignant, determine the stage of certain cancer, cell type or origin, study tissue development, study disease process, study cell death or cell repair. So all of them you can use IHC. But the question is how? And it's very easy and simple, by identify the epitope. Because when the tumor is malignant, it will contain certain protein which benign tumor don't have, and you can use the epitope from that protein to develop your primary antibody. And then you developed secondary antibody and stain your tissue slice. And same thing for the other applications. But first thing, you have to collect the tissue sample and make slice of them and stain them. And this week, we're also going to take a look at an image from a biosighting field neuron. And these images was already prepared, so I'm not going to show you the technique by itself. I'm just show you the slides. The technique by itself is very complicated, require really fine equipment. And this technique is a fiber tracing technique, which means we can trace the fibers of the neuron. So when we're preparing the slide, we have to make sure that the brain tissue that we collected is fresh, because this technique requires we to inject a dye into a neuron, and this dye has to be able to flow to fill up all over the neuron. If the tissue is fixed, the dye will not be able to go anywhere because the cell content is going to be solid, right? So that's why this technique will require fresh tissue. And then we are going to identify structures of a biosighting field pyramidal cell. So for a pyramidal cell, they all have a soma, and they will have a lot of dendrite, and they will have a axon, and then you will be surprised the axon is actually very small. And how do you tell if that is an axon or a piece of dendrite? So all the axons of all the pyramidal cells will have a axon terminal, which is this little guy here. And what is inside this axon terminal? That is neurotransmitters, right? And that is really how you identify an axon. If you don't see an axon terminal, it's probably a dendrite. Okay, and then you need to identify the apical dendrite and then basal dendrite. So apical means the surface or the tip. So in these situations, the apical dendrite will go to the surface of the brain, and then the basal dendrite will reach deeper of the brain. And how do you orientate yourself? You can zoom out and take a look at the image, or another quicker way to do it is telling from the direction of the axon. So the axon of these pyramidal cells will go into the deeper side of the brain. So the opposite side of the axon is going to be the apical dendrite, and then the same side of the dendrite is going to be the basal dendrite. Okay, that is the lecture part of this module, and I'll see you in the experiment part of this module. Bye bye.